So today we're very fortunate to be joined by Dora Costa, who is the Kenneth T. Sokulov Professor of Economic History at UCLA's Economics Department and a research associate at the NBER. Dora's research focuses on labor, demography, and health over the long span of American economic history, comparing and contrasting the past with the present. She has made important contributions across a range of topics of interest to our research policy network, including retirement, older age mortality, and morbidity and trends in population health. In her glittering research career to date, she's published in top journals in economics, economic history, demography, and social science, as well as publishing 2 books, including The Evolution of Retirement in American Economic History 1880 to 1990, which will certainly be of interest to our audience today. So today she's going to be talking to us about her recent working paper together with Lars Bigren, Benedict Graf, Martin Karlsson, and Joseph Price, The Economy, the Ghost in Your Gene, and the Escape from Premature Mortality. So with that, I'll hand over to Dora. You've got an hour to take it away.
Great. Thank you very much. I'm thrilled to be here. So if we think about the last 2 centuries, the 2 big achievements have been the miracle of economic growth and the increase in life expectancy. So one of the big concerns among economists, economic historians, has been what's the relationship between the economy's level of development and health? If we think about the past, it was a low productivity economy, a low income economy. There was crisis mortality prior to the 1780s. The work of Robert Fogel emphasized that chronic malnutrition was extremely important after the 1780s. So Fogel argued it's if you look at the heights and weights of people, you see that yes, they didn't die off in times of famine, but there were permanent sort of scars that were left on people, manifest in their height and weight. We can also think of the past as very much a low knowledge economy. People had no idea what to do about infectious disease and how to control it. So the work of Sam Preston, of Richard Eastelin, and of Angus Deaton has emphasized that the inability to control infectious disease was extremely important. And the gaining control of infectious disease is sort of a primary explanation for the improvement in longevity from 1870 to the 1960s, particularly for infant mortality, but also for mortality at very old ages. Now, one of the things which I am going to argue is that we have underestimated the role of early economic growth in the post-1870 improvements because we've ignored ancestral effects. And I'm going to argue that past and present economic growth is in the epigenome. What do I mean by this? What I'm going to argue is that we can see evidence of changes in gene function triggered by variants in food intake at critical ancestral ages, where critical ancestral ages, I'll discuss that in more detail later, are going to be during the slow-growing years. So one of the things which I think is new here is this emphasis on variance as a trigger. And furthermore, I think at least for prime age sort of men, so men between the ages, say, of 40 and 80, variance in their ancestors' food intake was as important as contemporaneous infectious disease declines. And this is for the cohorts that were born from 1830 to 1909. Now, why do we care about some of these distant processes? One reason might be these processes are still ongoing. if we think of developing countries today, there's a past history of famines. In many countries, the food supply is still highly variable. So think of Ghana with its rainy seasons, and in terms of past history of famines, India has had sort of a very long past history of famines. If we think about rich countries today, there are always situations such as wars. So if we think about variance in early economies, so you have both these lower income levels, but also higher variance. Why? Harvests were much more dependent on the weather. The food supply was much more dependent on local harvests. Ability to preserve food was much more limited. There were higher trade barriers. You had tariffs, interior tolls, transport costs were high, and you can think of the poor as just being extremely underinsured. When times were bad, there literally was nothing to trade. And one of the things which we can see, and I'm going to be looking at the case of Sweden in particular, and at least in the Swedish case, one of the things which you can see is there really was this discontinuity sort of roughly around 1840. After 1840, harvests become less dependent on the weather, for example. And one can see that also there are a lot of different changes which occur. So what do we think might happen in a low income and high variance economy? So we might think that it's Ole Bygren has argued that feasts and famines trigger an epigenetic change in gene function and that this change is then inherited. So cells go in and out of dormancy, and the outcome ends up being chronic disease for later generations, with men being particularly sensitive because one sort of plausible pathway is this is transmitted along the Y chromosome. So yes, we tend to think of the Y chromosome as not having so much on there, but it is still very important for some conditions. And if anyone sort of knows anything about epigenetics, we tend to think that, look, if conditions are good, these epigenetic marks should actually be replaced. But things may not function properly, particularly if you have one of these very poor economies. And if you just think of normal sort of pre-modern scarcity conditions, this might be sort of the place where epigenetic marks aren't erased in early gestation, and epigenetic marks aren't reprogrammed in late gestation. It could also be a place where you have in utero mortality selection. I'm not really going to be able to say much about that one, but again, this is sort of we think that these epigenetic marks should be replaced, but there's more and more evidence that the erasure doesn't happen and the reprogramming doesn't happen as should be expected. Now, we have pretty limited evidence on transmission in people. There's lots of evidence for plants, for worms, and for fruit flies. You can go up to at least 25 generations. But I'm not terribly interested in plants, worms, or fruit flies, and I'm not even that interested in mice. For mice, I think we maybe go up to 3 to 4 generations at most. Mice are relatively expensive relative to plants, worms, and fruit flies. So most of the evidence for people also focuses on feasts being bad and famines being good for descendants. It doesn't look at the variance. The samples have been relatively small in terms of not being able to look also within families. And there's also the bigger question of how much does it all matter at the population level? So I think for those interested in how we got from this very high mortality world, what's the explanation for the decline in premature mortality? I'm going to argue that variance is perhaps the unsung hero in the escape. Of course, it is not the only protagonist in the escape from premature mortality. I do not want to denigrate sort of the importance of chronic malnutrition and also of getting infectious disease under control as well. Those are extremely important. But I also want to introduce this third player, namely variance. So what is epigenetics? So one can think of genetics as just being a mutation in a DNA sequence, and this is then passed on and the phenotype changes. If we think of epigenetics, this would be a switching on and off of the gene function. And this could happen through DNA methylation. It could happen through various microRNA interactions. It could happen through various histone modifications as well. And this is an evolutionary adaptation to poor conditions, but then this adaptation is inherited. So why would we actually have this type of evolutionary adaptation? So Eva Jablonka has argued that one can think of epigenetics as sort of this precursor to long-term adaptation. We don't want to have adaptation sort of happening very quickly because conditions could change. Variance in the past was high. So before there's actually any type of phenotypic change, one can think you'd want to test the environment. What are some of the implications sort of this type of testing of the environment? One of the implications might be is that we may actually observe generational skipping in terms of inheritance. We might also sort of observe that there's more of a propagation of marks when the environment is stable. So one sort of other possibility is, when does transmission occur? It might concur when conditions are bad or maybe not sort of terrible, but sort of not so great, particularly at sort of some critical ages, especially in utero, where you have this reprogramming towards the end of gestation, or when you have sort of the erasure of marks early on in gestation. So one of the things which is going to be important for my identification is what happens if you think about how is the epigenetic effect passed along sex chromosomes. So grandfathers are going to have an XY chromosome. Grandmothers are going to have XX chromosomes. If we think about grandfathers, they're going to pass the Y chromosome to their sons. And you can see it just going straight down through the male line. If we think about grandmothers, they're going to be passing the X chromosome to their sons and then to their granddaughters. There are going to be other X chromosomes that come in there, but we can't really trace exactly sort of which X chromosome is coming from where. And if we looked at other lines, we're not really going to be able to do it. So the important thing is with paternal grandfathers, it's passed on directly to grandsons, but not to granddaughters. With paternal grandmothers and granddaughters, it's from paternal grandmothers and it's passed on to granddaughters, but not to grandsons. Now, one of the things which I will show is that we see these effects for paternal grandfathers and grandsons, but not for paternal grandmothers and granddaughters. And one potential explanation here is that the Y chromosome may be just much more likely to retain its epigenetic markings. So as you have this recombination during meiosis, it's with two sort of X's coming together, then you might be more likely to have more erasure rather than just an X and a Y coming together. One of the things which will also be important in terms of identification is going to be what are the epigenetically sensitive ages. And here we might think in utero is going to be one of them. The slow-growing years before puberty is going to be another one of them. And also the slow-growing years after puberty, but before adulthood is potentially another one. My focus is going to be on the slow-growing years before puberty. This is when you have, and this is something which has been pointed out in the literature as being sort of particularly important in terms of seeing some of this transmission. I'm not saying that in utero, of course, we do think might be also very important. So just think of the Dutch hunger winter studies. And in addition, the slow-growing years after puberty are also might be important. And I found this as in looking at Civil War, US Civil War POWs, where you do see this transmission. But here, what I'm going to be largely focused on is the slow-growing years before puberty. And this is where I do find in the data that you do see a lot of the action, though I don't really have sort of such accurate measures of the in utero period. So how do I actually test this? So one of the things which I'll be arguing is that it's the findings that I'm going to be presenting are going to be consistent with the epigenetics hypothesis. Obviously, it's one can't completely rule out other explanations. One can certainly kind of partially rule it out. Partially, it's going to depend, okay, what type of story are you going to find the most plausible? I will be able to rule in epigenetics. So there'll be a food shock to the paternal grandfathers in the slow-growing years before puberty, which affects the longevity of grandsons, but not of granddaughters. I'm going to be able to rule out socioeconomic transmission. There's going to be no evidence that the paternal grandfather's food shock in the slow-growing years before puberty affects his characteristics or that of his descendants. And here you can look at socioeconomic status, longevity, you can look at fertility, tried looking at sort of who's sort of the heir and might be inheriting, but controls for those, just it makes no difference. I'm also, another potential explanation is cultural or genetic transmission. Genetic transmission, we do think will take a while, so that seems to be unlikely. And in terms of cultural, I think it's going to be sort of a very convoluted explanation sort of for the transmission patterns that are observed. So yes, I'm looking at a past societies where it's there was much more emphasis on where you would expect that sons would be favored. But nonetheless, I mean, we don't find evidence of any type of extreme sex patterning in sort of the past in the Western world. So in the case of Sweden that I'm going to be looking at, it's who do the daughters marry? Well, they marry someone within the same social class as their fathers, sort of for the most part. So you do have kind of the same transmission of socioeconomic status across all lines. Now, I've mentioned before Sweden. Why am I looking at Sweden? Well, advantage of looking at Sweden is Sweden was pretty much on the periphery of viable agriculture. There were lots of harvest fluctuations, particularly in the far north. So you have advantages of remote areas. Sweden also provides good data in terms of recordings of sort of a harvest quality, as well as also good data in terms of availability of genealogical records, and also, and particularly sort of for some of these areas that were particularly subject to shocks. So in terms of harvest in Sweden, so one of the first steps of this paper was to create sort of harvest data series by Swedish counties. And this is based on sort of a combination of data from Helstenius, from Statistics Sweden, sort of various published records from Utterström, and from Hallberg et al., looking at tithing, where you do have evidence of sort of, well, what happened to harvests in the past. And basically, what the classification that we're going to be using is going to be just good, normal, and bad. So it is sort of a very much kind of, it is very broad, but given that we're using sort of these very different series that have been put together, I don't think we can really get to a much finer level. Now, what is going to be a harvest swing? It's going to be sort of this radical change. So either you go from a good to a bad harvest, or you go from a bad to a good harvest from one year to the other. And here, what I do show later on is that it doesn't matter which way the harvest swing goes. It can go from good to bad or bad to good in one year or another. The sort of the results are basically the same. And I should point out that this is not a balanced panel. You're much more likely to have northern Sweden early on. And I should also point out that, yes, there's certain judgment calls that were made in terms of sort of assessing these harvests as good, normal, and bad. However, if you look at sort of these harvest measurements and compare it and look at sort of various sort of contemporaneous predictors, they do appear to make sense, fortunately. So big advantage of Sweden, I guess it's before they had the nanny state, they had the nanny church, and it was the nanny church, it was the local parish priests who were collecting harvest statistics, which they then compiled and shipped off to Stockholm. So one of the things which you observe is that there are more shocks, and they're bigger shocks prior to 1840. So most harvest swings were before 1840. So here, this is the fraction of years with any radical harvest change. So this is a map of Sweden. Norway is towards the left. It's Finland is towards the right. It's and up above you are basically sort of and up above you, yes, you are getting very close to the Arctic Circle. So note that it's early on, you do have many more harvest swings. They are concentrated sort of further in the north, but you still, and after 1840, you do have fewer harvest swings. A caveat, of course, is this is only looking at the really big swings, but certainly one possibility, which I can't discount, is that maybe it's some of the smaller swings would have similar effects to what I'm finding also. In terms of do these things do these harvest measures make sense? If you look at sort of before versus after 1840, a poor harvest was more likely to follow a poor harvest. One of the things that this suggests is during a poor harvest, people were probably eating their seed grain. So you do end up having this persistence of bad conditions. Bad harvests were also much more likely when there was drought. And after 1840, this changes. Drought no longer becomes as important. And this is with the introduction of more drought-resistant varieties. And prices were more responsive to bad harvests. If you look at the period prior to 1840 rather than after 1840. And in addition, it's if you formally sort of test for market integration, looking at sigma convergence, doing a principal components analysis, yes, there was less market integration before 1840 versus after 1840, as one might expect. Why do you have some of these changes? After 1840, farmers were incentivized to produce surpluses. You have abolishment of export regulation, of interior tolls, of price controlled, transport cost fall. It's cities and non-agricultural areas become good markets for farmers. So there's more sort of an incentive to produce, and you have more market integration. And so if this is just the coefficient of variation of the barley price, so barley was particularly important in northern areas, and you can see, yes, there was this decline. So given these changes with 1840, I'm going to focus on the descendants of grandparents born before 1820. There are going to be two data sets that are used. One is family search genealogies, and the other are parish reconstitutions that were done for selected areas of Sweden. And this is sort of the demographic database at Umeå University. So I'm going to start by talking about the family search data set first and present those results first, and then I'll be presenting results from the demographic database. If we think about sort of this is sort of the primary sample with median birth and death year, and you have 4,783 men, 4,453 women. They all live to age 40. Why age 40? I'm interested in looking at older age mortality. So this was sort of kind of arbitrarily sort of picked, just, okay, let's look at age 40, particularly sort of for these past populations. And everyone in the sample was born and died in Sweden. And this is a case where I can actually extend the sample to look at those sort of who left Sweden and immigrated. The results are not as strong sort of for that population, probably because conditions sort of were changing so much for those who immigrated, but you can still see sort of some effects in those populations. So everyone here, so the median age of birth is roughly 1862, and these are folks who died in 1933. If you're thinking about born in 1830, died in 1900, sort of for the mothers, the fathers were born roughly in 1828, and the paternal grandfathers were born roughly in 1794. So this is a sample where I have everyone was born before 1820. There's harvest information is available on all four grandparents at ages 0 to 20. And Gotland, which is tiny island, Nordbotten and Västerbotten, the last two are both in the very far north, are overrepresented. The sample has been linked to the Swedish census, where I can link sort of where we can link the children, and you can actually get sort of a socioeconomic index using machine learning prediction. And in addition, one can also link to contemporaneous infant mortality rates by county. Now, I think one of the things I should point out with this sample, this is a genealogical sample. It is based on Mormon genealogies. However, if we think about sort of the areas that are overrepresented, these were not sort of big areas that drew sort of converts to the Mormon church at all. So I think a lot of this is just sort of basically with family search, just ability to grab sort of genealogies from various places. What is going to be the main specification? Identification is going to come from grandparents' harvest conditions in county of residence at critical ages. So basically, this is going to be sort of a birth year times birth county fixed effect for grandparents. I'm going to be treating each year of life T between age 40 and 80 for individual I as a single observation. You either live or die in that year. So the dependent variable is equal to a 1 if died in year T, 0 otherwise. And I'll be estimating linear probability models for each sex clustered on I. Why linear probability models? This is going to allow for fixed effects, so age, birth year, grandparents' birth years, and birth counties. So the main specification is going to be sort of, did you live or die in a specific year? As a function of, did the grandparents experience a harvest swing during a particular age group, where A sub IT is and in addition, I'm going to be controlling for fixed effect for age, of course, fixed effect for own birth year, and fixed effects for grandparental birth year, birth county, and I'll be clustering on the individual as well. So if a grandparental harvest swing does not affect the grandparent's health or socioeconomic status, this actually has a causal interpretation. Later on, I'll be adding controls for, was there a bad harvest in utero? What about contemporaneous infant mortality by county? What about socioeconomic status, parents' longevity, etc. So the explanation is going to be just what is left standing. So epigenetics is going to predict that it's grandsons, but not granddaughters' longevity that will be affected if we observe a paternal grandfather's harvest swing at any time between the ages of 9 to 12. And one might think that there are going to also be no effects outside of any critical grandfather ages. There's going to be really no predictions about effects across generations because of this possibility of skipping. So socioeconomic status, it's going to require that there is sort of, you would expect to see effects on grandsons' socioeconomic status and they're also their wives' longevity, since the wives are in the same part of the family. And this is also here, it's one should expect that there will be no skipping. It's one of the things which also we should be able to see is If we think that socioeconomic status affects sons' health and therefore the health of grandsons, then sons and grandsons' longevity is going to be affected, but again, sort of no generational skipping. We might also think there are fertility effects. We don't find any fertility effects at all. So what is sort of one of the primary results? Well, if you look at this sort of graphically, and these are looking at does among sort of men at the top, it's men, and if you look at the father's father, did the father's father experience any radical change at specific ages? So if you look at sort of ages 0 to 2, 3 to 5, 5 to 8, 13 to 16, 17 to 19, yeah, overall, you don't really see anything. If you look at the ages 9 to 12, increase sort of the change in the death probability would be 0.004. If you then, and this translates into roughly half a year of life expectancy at age 40. If you look at agricultural counties, then this increases to 0.006 or 3/4 of a year of life expectancy at age 40. If you look at any sort of radical change in agricultural counties and parishes, this goes up to 0.009, and you lose 1 year of life expectancy at age 40. With women, you don't really see any of the same type of pattern. If you look at other lines, there's nothing for women and fathers' mothers. If you look at men and fathers' mothers at ages 17 to 19, yeah, maybe there's something there. If you look at men and mothers' mothers at ages 8 to 10, yeah, maybe something there. But what actually can withstand multiple hypothesis testing? Basically, I have different regressions by sex. Within each regression, there's 6 age groups and 4 grandparents. In addition, there's separate regressions for agricultural versus non-agricultural counties. And further, you can sort of divide it up by parish. So ultimately, it's only sort of this father's father, ages 9 to 12 effect for men remains statistically significant once you account that in the extreme, there are 102 multiple hypotheses. Now, we also looked at other ways to classify counties, trying to look at the extent of market integration. That sort of led to slightly smaller magnitudes than looking at the effects of agriculture alone. But it was sort of this smaller set of sort of counties that aren't kind of in the integrated set. So what this maybe suggests to me is it's not just market integration, but it is sort of this agricultural dependency of small farmers on very volatile harvests. So what this table shows is that as you add controls, so what's the time varying log of infant mortality? If you add an index for socioeconomic status, if you just add sort of the HISCAM in there, if you add father's longevity, if you add the mother's longevity, it's yes, these things do matter, but ultimately it's the original magnitudes, and bad harvest in utero, of course. You can add these controls, but the original magnitudes remain sort of unchanged. If anything, there's a slight increase in the original magnitudes. So getting some sense of how do we compare sort of the magnitude of ancestral effects for men. So if you look at sort of the father's father radical change at ages 9 to 12, increase in probability of dying in any year of 0.005. A bad harvest in utero, increase in probability of dying of 0.004. If you think about a half standard deviation increase in the log of infant mortality rate, sort of by county, increase of 0.001. If you think about sort of change in half standard deviation increase in the change in the socioeconomic index, 0.001 in terms of absolute magnitude. So sort of this father's father radical change effect does become important. Is sort of in terms of magnitude, it does compare sort of favorably with things we care about, such as conditions in utero. One of the things which we should also expect is that there is going to be persistence if conditions are sort of bad. And what we do observe is stronger effects if both the father and the son were born in the winter. Why the winter? Food supplies were at their lowest. People are living off of the previous harvest, which would come in sort of in the fall. And in addition, it's sort of there's nothing fresh that's available, which would provide vitamins. So one of possibility this raises is that what may be affected is the reprogramming of the epigenome. If we think about, so the show sort of results for the semester of birth and paternal grandfather's radical change. So you can see if the son is born in the first semester and his father is born in the first semester, you're going up to 0.008. And if the son is born in the second semester and the father's born in the first semester, it's point increase in probability of dying in any year of 0.004. Now, let me now move to sort of the painful kind of family reconstitution data set. So this is, and here what I'm going to be doing is using within family identification. So there are going to be two grandfathers who are brothers. One grandfather experiences a food shock, and the other grandfather does not experience a food shock. So let's just say it's the food shock is at ages 9 to 12. And here in the set of ages 9 to 12, so there's 678 grandfathers, they go down to the fathers, and then you can trace down sort of the different lines from grandfather to father to son, and also look at the granddaughters as well. And so here, sort of for the ages 9 to 12, there are going to be 2,520 grandsons. And to ensure it's all the same, they're all going to be born in the same parish with a median birth year of roughly 1865. And one of the limitations, of course, of family reconstitution data is you are looking at very much selected parishes. So there's a predominance of 3 parishes in Västerbotten County. And these data have been linked to the Swedish Death Book to obtain additional and also national death information. So yes, everyone's born in the same parish, but they can migrate among the grandsons, but they can migrate later and they can die elsewhere. So here, controlling for there are going to be individual regressions, sort of controlling for different sort of age groups, and where one grandfather receives a shock, the other one doesn't. So for example, it's very simple specification, say controlling only sort of for a limited set of variables, including the infant mortality sort of in the county. And this is going to be just repeated sort of for various other age groups. I can do this with the family search data also, but it's very small sample, and it does give me much stronger results than in the original family search data. But these are results from the parish reconstitution data. And so here you'll note, and this is looking at all ages, given the sample size, rather than looking at ages sort of just 40 to 80 in terms of mortality, focusing there. It's sort of everywhere. It's all ages throughout sort of the life cycle. And here you'll note this is men, this is women down below. If you look at sort of men, it's sort of a increase in probability of dying at any year of life, starting at age 40 until death, increase of 0.005, and this translates to roughly 1.3 years of life. For women, sort of, yeah, maybe slight effect there, but you really don't see much going on there, though somewhat sort of surprising, sort of this 13 to 16 here, which you also see for men as well. But what actually here, again, there are 12 hypotheses, what actually stands up, and only radical change at the father's father ages 9 to 12 is statistically significant. There are various alternative explanations, of course, but we find no evidence for socioeconomic transmission. There's no effect of the paternal grandfather's harvest swing on the longevity of sons or daughters, on the longevity of wives or grandsons. It's on any sort of socioeconomic index. There's no extreme sex patterning and socioeconomic transmission in Sweden in this time period. There are no fertility effects. And there are also no air effects. As I mentioned before, the direction of the harvest swing doesn't matter. And if anything, what one can observe is that sort of this ancestral yearly harvest swing is worse than having, say, a time period at the paternal grandfather's ages 9 to 12 of bad and all normal harvests, or of having mixed harvests and no yearly swings. What are some additional findings? There are no effects if the grandfathers were born in 1820 or later. And you don't actually find sort of any evidence in terms of mortality at younger ages. As I mentioned, if you expanded sort of a family search sort of to anyone, even to those born abroad with the paternal grandfather, as long as there are controls for the father's father, sort of a fixed effect, so paternal great-grandparents, it's fixed effects, there are results. Similarly, in the demographic database, if you look at the cross-section, there are sort of results once you control for the paternal great-grandfather's fixed effects. And why might some of these sort of fixed effects matter? Suspect that if a gene is going to be turned off and on, you actually need to have sort of some inheritance of that gene. So what's the likely population impact here? So this sort of kind of lays out sort of a potential sort of scenario, looking at different birth cohorts. So starting, say, with the birth cohort born from 1830 to 49, and then going through the birth cohort born from 1900 to 1909. So just looking at male life expectancy at age 40, it's you're going from 29.8 years to 34.89 years at the end of this time period. Now, if we think about what's the fraction of paternal grandfathers who experienced any sort of radical change at ages 9 to 12, you'll note you're starting from 0.297, but then sort of the cohort that's from born in 1860, 69, had paternal grandparents that were particularly hard hit. Why? This is going to be, the assumption here is sort of like roughly 30 years sort of separate the generations. This is going to be the generation that was hit particularly hard by sort of all these series of bad harvests around the Napoleonic Wars. So this was a particularly tough time sort of for Europe. In addition, you also have sort of exploding volcanoes too in this time period as well. I can't go earlier. The reason there are asterisks here is because I don't sort of, these fractions, it's not available for all counties in Sweden. And this is sort of an estimate of the life expectancy if we could abolish all radical change. What would be the gain in life expectancy? So note sort of increase in gain in life expectancy going up to 1.37 years, and then a decline as you get into the later cohorts. The other sort of thing, what one can contrast with is suppose that life expectancy at each age, sort of starting at age 40, that the infant mortality was the infant mortality sort of in the next 10 years. And infant mortality here is just a proxy sort of for how bad the infectious disease environment was likely to be. And one of the things which one notes, of course, is over time, you do have these big changes in infant mortality. So over time, sort of the life expectancy gain from infant mortality sort of changes. So from infectious disease becomes more and more important. But nonetheless, it's at least sort of for some generations, it's these reducing sort of this harvest variability was as important as reducing infectious disease rates. So one of the questions is, how does one actually reduce harvest swings? I mentioned before, sort of if you think about the history of Sweden, it's sort of the incentives and institutions changed to allow farmers to keep more of their produce. They were more incentivized. Export regulations were improved. There were interior tolls. Farmers, so Sweden went from being sort of a grain importing economy to being a grain exporting economy. As farmers sort of are able to keep more of their surpluses, people are moving out of agriculture, and you have sort of you're no longer as dependent on agriculture. Technological change, of course, is very important. Transport costs, it's fall. And in the case of Sweden, I should point out that it's not until sort of the early, really 1870s that some of the very far areas of the north were actually start to be linked by rail. So in winter, a lot of these areas were shut down, particularly when the Bering Sea sort of would ice over. And you have also better crops as well. This is going to be a slow process. It's hard to find sort of this sharp discontinuity in when you suddenly get all of this technological change, but eventually you do get this process that is going on. So I think one of the things kind of in terms of looking at sort of today in terms of developing countries, certainly one of the implications would be how can one sort of ensure that there is sort of this reduction in variance and that we can effectively protect against variance to try and avoid these harvest swings. Another lesson, I think, of course, is in terms of transmission of epigenetic marks, so improvements in conditions may well matter. So Ole Bygren has argued that we we should probably expect to see sort of maybe up until the fourth generation, there might be some effects. It's in the samples that we use here, we can't really go up to 4 generations, but Ola actually has a very small sample, which is different, where one actually can sort of observe effects for calyx and also in a different sample, sort of from Westerbotten, where you have fourth generation effects. So how much kind of is epigenetics here? It's the evidence is consistent with epigenetics, but of course, one of the things sort of one should be aware of is it's super hard to know. Is it epigenetics or is it something else? So I think sort of some modesty is in order, but nonetheless, sort of I think one of the things that we have shown is, yes, there is this strong effect. And this is not just sort of a hypothetical sort of a situation. People now are actually looking at how can we actually use epigenetics directly to improve human health. So if you think about sort of people with genetic conditions, if you go in and try and actually edit the genome, that's a brutal process. And surviving it is sort of a little bit of a question mark. So what researchers are looking at is how do we actually switch some of these signals on or off? And there's a question of sort of what type of what can you actually do to change sort of these signals? In some cases, it's certainly at the simplest level. What one can do is prescriptions such as exercise, are certainly help, but there's also the question of could actually scientists eventually directly intervene to make changes? So I'm going to stop here since sort of the rest is in the appendix, but here I'm kind of going to leave sort of a kind of this final sort of a graph. So this sort of repeats sort of what I showed before. But if you look at sort of non-agricultural counties and parishes, you can see no effects there also.
Thank you very much, Dora, for a really interesting, fascinating presentation. So to get started with the questions, I think just actually leading right off what you were saying at the end there, we had a question from Tiernan Evans here asking, I mean, to what extent modern genetics, how far away we are from being able to identify specific mutations and or specific markers and their outcomes, and is there perhaps a way that studies like this and looking at shocks like this might actually be useful going in the other direction and being able to identify the specific markers. I mean, you could probably tell from the words I'm using here, I'm absolutely not an expert on epigenetics, but the specific biological markers that perhaps are driving these sorts of effects by kind of reverse engineering them from something like this.
Yeah, so I think sort of in a sense, sort of the holy grail, I think is always going to be direct biological tests. I've discovered that the geneticist and the epigeneticist fight like cats and dogs. Sort of the geneticists sort of want kind of almost this complete identification where you could actually observe things across all the generations and actually observe sort of literally changes in sort of the marks. Kind of, I think the closest that we've gotten in human populations is looking at the in utero period with the Dutch hunger winter studies, where one can observe sort of these changes in DNA methylation. More so, yes, one can sort of observe that, okay, there are kind of these markings which might matter. I think kind of a question then is, how do you then go about and literally change these? And what, and sort of, and also the other question is which markers sort of are more and more important. So there's arguments kind of, yes, DNA methylation is the cheapest sort of to measure. So there are a lot of measurements there, but people are also now looking a lot at microRNA. And how quick sort of are sort of these changes? I think one of the things that sort of I think is come out of this is sort of its fathers are sort of particularly important, kind of including sort of the behavior of fathers. So it's bench scientists kind of run all of these experiments where they'll subject sort of undergrads to, they'll stuff them full of sugar and they'll observe changes in a sperm. So I think a question is, are these changes then transmitted later on? Obviously we do not want to do these experiments on people. So I think this is where sort of historical studies can help, and they can sort of provide sort of some clues about what conditions should we actually look like, what conditions should we actually look at, what ages should we particularly look at in terms of critical time periods. It's what type of triggers also might be particularly important.
Thanks. That kind of speaks to another question we had here from Kotha Hayat, who was asking, it kind of addressed it there, if there are current means to reverse epigenetic effects, e.g. through methylation. So I think you kind of touched on that. I don't know if there's anything else you wanted to add there, but there's another question that I think also links to that a little bit, which is, I mean, you talked a little bit about alternative mechanisms, things like social, cultural norms, particularly on that sex-specific patterns. So I guess what part of the question there is, to what extent is this Y chromosome male lineage effect settled science? Is that really kind of an accepted for our audience, so they're not necessarily experts in this technical field? To what extent is that really settled science? And are there other mechanisms that could produce that sex-specific patterns, and or more generally the kind of other transmission mechanisms that you mentioned briefly in the talk, but didn't necessarily have time to go into in detail, that wouldn't need an epigenetic framing?
Yeah, so if you look at experiments on mice, and with the caveat that we're not mice, and sort of one of the points about epigenetics is one does need to be careful about drawing sort of conclusions from different species, just because you have very different sort of at birth processes in terms of sort of what might be transmitted sort of across the placenta, etc. But with with various studies that have been done on mice, so particularly sort of with food stress, it looks like sort of effects do tend to be stronger if you go through to the male line in terms of, so males do seem to be more sensitive if you look at mice. So again, preponderance of evidence sort of with mice, we're dealing maybe with samples of 48 or so, given the cost of mice. And of course, laboratory mice are very different from field mice. I think sort of one of the issues, I think so, in terms of sort of could it be socioeconomics or anything else, sort of we've looked at sort of trying control for who's the heir. We've sort of tried to control for sort of various measures of socioeconomic status. It doesn't seem to matter. I mean, one of the reasons I think that sort of there aren't big effects on socioeconomic status in part is because this is an older age sort of phenomenon. By the time you get to socioeconomic status, it's already been established. So the types of conditions that have been implicated sort of in epigenetics would be sort of various types of cancers, but particularly, I think sort of stomach and colon cancers in particular as being sort of particularly epigenetically sensitive. The other types of conditions that have been implicated as well would be sort of a type 2 diabetes, for example, and sort of kind of all sort of the constellation of factors that are related to that. And there too, one can think of, it's like, well, okay, maybe you inherit this gene that predisposes you to type 2 diabetes, but then there's a question of how do you switch it on or off? And here one can think sort of interventions such as diet, sort of exercise, etc. But can we actually directly go in and sort of change it? Well, we probably won't be experimenting with type 2 diabetes, but we'll probably at some point start to be looking at conditions such that are sort of where there aren't sort of any other types of interventions that are available. And those still are very much kind of in the animal experiment types of stages.
Mhm. Thanks. So switching track a little bit to more like the technical identification. So we've got a couple of questions here. One from Siavash Mahades asking to what extent you're identifying assumptions are relying on the assumption that Sweden was consuming only locally harvested food, particularly in the context of grain trade with Baltic areas. Apparently being a major factor in this period, and I think there's a broader question there as well around the harvest definition, um, and how important the extreme swings are there, and more broadly, what motivated you to construct the harvest index in the way that you have done, with the kind of good and bad swings rather than, I mean, you didn't have time to go into the talk, but presumably there is some underlying harvest data that's more continuous underneath that, and to what extent those big swings are important and what motivates looking at those big swings rather than some more continuous measure.
Yeah, no. So unfortunately, it's there aren't sort of good continuous measures. It's because for a lot of this time period, it's the data simply are missing, and so that was part of the motivation. The other motivation is Ole Bygren, actually. So he has a paper that was looking at so I guess I should say two papers. One was looking in utero, and this was using an early version of the DDB data, where what he found is if there's variance in utero in terms of the harvest quality, that was what was extremely important for understanding later mortality. And his other paper was actually looking at paternal grandmothers, and he found that sort of, I think between the ages of 60 and 80, there actually is an effect of sort of the paternal grandmother being subject on the granddaughters of being subject to a harvest swing if you look at certain specific conditions. So sort of kind of the sort of a stroke type of conditions, sort of in terms of sort of and cardiovascular diseases sort of kind of related to that. So there's some evidence sort of already that was sort of about harvest swings. But the other reason sort of for looking at harvest swings was just it was sort of puzzling. So one of the things which I found puzzling sort of about the previous literature is sort of this, and it certainly is possible, I don't want to exclude it, is that you could look at what some of the previous literature found is that it's if you look at sort of these paternal grandfather effects and grandsons would actually want a paternal grandfather who actually experienced sort of a poor harvest. And it's if he experienced a good harvest, that was actually bad. So that was sort of a little bit puzzling. And one possibility sort of that raised, and which previous sort of researchers really didn't have sort of a big enough sample to actually test, is maybe it's not sort of the quality of the harvest itself, but maybe it's actually it's actually related to sort of the variance in the harvest that matters. So that's one of the things which led to it. Could this be more continuous? I mean, I think certainly there's a paper. It's by various co-authors, including Denis Vaguero, which looks at the Sweden sort of from roughly 1870 down to the present, looking at Uppsala. And there they have sort of more of a 7-point sort of a harvest scale, because then later sort of Sweden started to produce sort of a finer sort of a harvest measures in a sense. So that's sort of what led to kind of the move there. I should point out one of the things which I did try to do is actually predict some of these harvests, but sort of just using sort of because I was hoping to say get an instrumental variable to just for potential measurement error, trying to look at sort of looking at temperature measures, but that wasn't actually, it was actually very hard, and there were sort of some weird also negative correlations in terms of predictions later on. So sort of kind of abandoned that. I think one of the problems is that, yes, it is sort of good versus bad or good versus normal, normal versus good. It is a bit of a knife's edge type of thing. So that is sort of a potential sort of a worry. But it does, I don't think that I can actually do much better in terms of these things. Because often with the historical data, it might actually be sort of a very scanty evidence. It's just sort of this brief statement about what it is. So if we take a time period sort of just out of the sample that I'm using, say 1868, if you look at the published records, it's sort of right after sort of the famine in Västerbotten, sort of the description is it's a small harvest, but it's a good harvest. It's so what does one make of that? I think most people, so kind of the previous sort of measurements just put down that that is a normal harvest. Well, maybe not. If small is something that matters, then that might matter. But it kind of, there are definitely, I think, limitations to some of these measures, but it is the best, I think, that can be done if one wishes to have this long data series, which I think is necessary just to use as much of the data as possible.
I
So
see.
I
Thanks
can't
so much.
remember if I've answered
Yeah, no,
the
so
full
I think I
questions.
was trying to be too efficient and merge two questions in one. But so the first question was about the assumption that Sweden is consuming only locally harvested food,
Um,
particularly in the context of grain trade with the Baltic areas. To what extent that's something we should think about here?
Yeah, no. So it certainly was sort of a consuming sort of, but I think sort of the primary place where you are going to be getting this, of course, is going to be Stockholm. There are going to be certain areas in the far north where you're just not getting it. Just market integration is just not that high. And ultimately, if you think about, yes, there were attempts sort of made to have various granaries, etc. But if you think of this just from the poor farmers sort of a point of view, it's when times are good, it's so there's all of this sort of last year's grain that's sort of floating around. It's not as high quality. You can't get as good of a price. It's when times are bad, though, even there, you need to worry about, has the grain been stored properly? There's risk of fire, risk of vermin, etc. So there are lots of risks. In terms of the Baltic trade, I can't remember which particular areas. I think Gotland was also one area that probably would have been big on the Baltic trade. Um, One of the things which we tried doing sort of in the family search data is restricting this sort of by different areas. So particularly because there is sort of this preponderance of Västerbotten, Norrbotten, and also Gotland, which actually, so Västerbotten and Norrbotten in sort of the measures were definitely not that well integrated. But even sort of excluding those Västerbotten and Norrbotten, we still observe these effects in terms of what happened, in terms of the actual radical change and grandfather's radical change and men. So there are effects still which are there, and it's not just sort of the least integrated areas that are affected. So one does see this elsewhere. But yes, of course, there was trade, but it still is limited. And particularly with remote areas, it's if there is an emergency, how do you actually get foodstuffs up there? Particularly when you have transport was super expensive. It's seas are frozen over, and you certainly wouldn't want to. And the North Sea also was extremely treacherous. So even sort of where you don't have waters frozen over, you really didn't want to cross the North Sea in winter.
I see, thanks. That's very clear. So sticking with the more technical questions before we broaden out a little bit. So I think if you could talk a little bit more about the genealogy data you have, like what the coverage, I mean, overall the sample compared to the Swedish population is obviously quite small, but what that coverage is like, and if there's maybe a risk of some selection or survivorship biases there, maybe linked to migration effects, that migration is more likely in areas that have a lot of harvest variability or something like that.
Yeah. So I think actually, I think so the far north sort of areas, these were not kind of the big migrant sending areas. Now, in terms of so there's the family search genealogy data, and of course, one of the drawbacks of using sort of these family genealogies is it is going to be sort of the survivors who are putting together the genealogies, though in some of the data that are in there, that's clearly not the case. It does look like individuals just went through and decided to fill out genealogies because some of these things just end. So I think one of the advantages, so I think the only way one can kind of get around some of that survivorship of who puts together bias in terms of who puts together the genealogies is to look at the parish reconstitution data. In terms of the migration of who migrates, well, one of the things which one can, so with the, in terms of the family search data, it's I did the restriction in the results I show to sort of born and died in Sweden. Part of this was to also be able to control for contemporaneous infant mortality, et cetera, and also just to be able to get better controls for Swedish conditions. But in those data, people could have migrated anywhere as long as sort of whoever put together the genealogy was able to effectively follow them. And if you look, say, at and you do observe a lot of migration to the US, but also to other parts of Europe as well. And in the case of the family search data, yes, there is this, if you look at the US migrants, there are areas that are overrepresented, not too surprisingly, are Utah and Minnesota. Nonetheless, I do think that it's one can. So yes, there is this bias, and I do think that one of the questions is if we want to find these effects, where do we look at? Ideally, we'd like to keep conditions sort of as similar as possible. Once people migrate, they are going to start to acquire sort of more of the characteristics of the people sort of they migrated with. So you are going to have kind of this change in conditions. So it's not sort of the laboratory, conditions are going to be changing. And I just can't really control for some of these changes in conditions. But what's sort of stuffed kind of in sort of one of the appendices, I believe, is just sort of some results from for family search, looking at the full data set, kind of including at all the people who migrated sort of elsewhere, where the elsewhere is particularly to the US. But most of the migration to the US actually was from the areas that did not experience sort of a, I mean, maybe with the exception of Jamtland, which is not that well represented in the family search data, it tended to be sort of from areas that did not experience sort of enormous kind of harvest swings. It wasn't the far north.
Thanks.
Now, with the family search data, if anyone is interested in using it, I should put in a pitch. It's Joe Price is super eager to sort of to be helpful for any use of family search data.
Oh, that's very good to know. So thinking a bit more about the bigger picture now, so policy implications and the kind of relevance for today of your findings, yeah, where do you see these as being greatest along two dimensions there? So in terms of developing countries, where there's maybe a direct parallel with climate shocks hitting harvests and those sorts of things. But is there maybe also applicability today in rich countries through this channel or maybe through other epigenetic channels? And then the other question there is around policy timelines. So we're talking about multiple generations before these things are having, or before the measurable effects that you're measuring are being picked up. So how should a policymaker think in that context of these potentially generational timelines from cause to effect?
Yeah. So I originally sort of came to this sort of with my interest in Civil War POWs, where you could see sort of effects of US Civil War grandfathers kind of at roughly sort of ages 18 to 24, but not at older ages, and these trace down to the male line grandson. So originally I was interested in that, and I was sort of more interested kind of in these sort of ages sort of 17 kind of to 19 effects. I'm not seeing anything in these data sort of around sort of these ages. One possibility is that sort of outside why there are no effects here is precisely probably the migration thing. And here, in terms of, you might expect sort of young men, they're going to be the ones migrating. Children aren't going to be the ones who are going to be migrating. And if anything, maybe it's the fathers who might be going off somewhere to search for work, but I think it's more likely it's just going to be the young men who are going to be leaving. So that's probably why there are no effects there. But in terms of a more modern population, I think one of the things which I am interested in is, say, are there effects among World War II POWs, particularly ones who were held in horrific conditions? So it might be, say, among sort of the POWs who were held by the Japanese, for example, where you did have these much more horrific conditions. And can we then trace what happened? We know what happened to them, but there's a question of, well, what about their children and what about their grandchildren? So there are always going to be these populations. Other sort of interest is, and there's been research on the World War I German famine, where sort of going down to sort of the first generation, you're not finding so the effects that you can look at are kind of limited just because of sort of the ages, but there are definitely sort of some mental health effects that show up. So there's a question there too, kind of, well, maybe these are sort of some lasting effects. And what about World War II, of course? Well, we've seen sort of these Dutch hunger winter effects. Of course, with developing countries, yes, there is sort of much more of this history sort of in the past, where a much larger proportion of the population is going to be affected. So there's a question, I guess, sort of for policymakers, what can we actually do? You can't go back and change the past. So I think the only thing you can do is just try and look at what actually can help reverse some of these epigenetic conditions, and among which groups sort of are some of these epigenetic conditions, where do they manifest? So for example, kind of in these data, we saw that with a semester of birth, that was actually one of the important interactions. So one certainly policy prescription might be if you are worried about this, so particularly for people who might have sort of these genetic markings, sort of a good maternal nutrition is going to be very important. And of course, we know that on its own, good maternal nutrition has positive effects. But this is just sort of another way kind of to highlight that, yes, this is going to be important. We actually want to do what we can to prevent these marks from being transmitted. Now, will these marks not permanently be transmitted? We don't know. Maybe these marks will just be dormant, and if conditions become bad, they'll pop up again. We don't know.
Thanks. And so I think we've got time for one final question before we need to start wrapping up. And so your paper so far was really focusing on mortality, but I wonder if it speaks at all to other outcomes like morbidity. Is there any sense in which there is a health effect here? Or perhaps, I mean, you've talked a bit about socioeconomics not driving these results, but I'd be very surprised if there aren't some sort of socioeconomic effects if there is a health channel going through here. So is that something that your paper speaks to at all? Or if not, maybe I can invite you to speculate as to whether there might be broader effects on things like morbidity, health, and economic outcomes.
Yeah, yeah. No, I think in terms of sort of the economic outcomes, and I was observing this also with US Civil War POWs, I think one of the reasons when it's not observed is just at what ages. So it's by the time that sort of these men are observed, these are sort of chronic conditions at older ages. Socioeconomic status is pretty much well developed by then.
Mhm.
And I don't have, say, like more refined measures about how did granddad actually kind of do in his old age, etc. At least sort of, I mean, everyone kind of was sort of so these are populations where we still don't have sort of a really fine measures of socioeconomic status.
Mhm.
It's but I think here it kind of comes down to it's when do these conditions manifest themselves? I don't have information on morbidity, unfortunately. I don't think that the morbidity would manifest itself because if we think about various types of cancers, type 1, type 2 diabetes that have been implemented with epigenetic changes, and this is going to be in other research with smaller samples, so in Ulla Bygren's over-calyx studies, in Denny Vaguero's Uppsala studies, then in those cases, it really are these chronic conditions, which most people fortunately do not get when they are young, but which do manifest at older ages. Where you might see some manifestation might be in things such as, say, measures of overweight, for example, but I don't think that sort of any of these overweight measures are kind of big enough in terms of actually having an impact on socioeconomic status. And unfortunately, I don't have overweight here.
Okay, great. Well, thanks very much. So we're at time now, so unfortunately we'll have to wrap up. So it just leaves for me to thank Dora very much for your time and sharing your research with us today, and thanks to all the attendees that joined and participated as well. I'd also like to thank very much Megan Gabriel and Imi Scott for all their work behind the scenes on the technical side and the logistical side, setting all of this up, and of course to Andrew Scott, the leader of the Research Policy Network. Just finally, before we leave, a couple of points to mention. The recording of this webinar will be available on the RPN website in the coming days. You can also watch the video of our first webinar there with Amy Finkelstein there. That's still available there as well. And finally, our next session will be on the 19th of May, where Darren Asamoglu will be talking to us about his work on fertility and economic growth. So Thanks again to you, Dora, for talking with us about your research today. And thanks again for everyone for joining us.
Great. Thank you.